Hi, this is Miss Litton, and this is 7th period review. These are excellent APBio students. Say hi. Hi. I would like to, as we review this chapter, to address potential essays that you could have over this chapter as well. Does that sound like a good idea? Yes? Yeah. Are you surviving this day? Yes, you're surviving this day. All right, so the first thing, remember, there were 3 big ticket items, okay? One was the cell cycle, the regulation of the cell cycle, controls in the cell cycle that we talked about. Another thing we talked about was focusing on the steps, obviously, of interphase of the cell cycle, and then the steps of mitosis and cytokinesis of the cell cycle, and kind of understanding the dance of the chromosomes. And then the last thing we talked about was when it goes wrong, when you lose control in the cell cycle and have something like cancer. And actually, that was the second to last thing. The very last thing we talked about was prokaryotic cells. And do prokaryotic cells do mitosis?
Yeah.
What kind of chromosome do prokaryotic cells have? Single circular chromosome. Do they have linear chromosomes that line up or do any of that? No. Okay, they do not. Okay? Do you remember what it is called for prokaryotic cells? Yes, binary fission right here. Remember that? It's a single circular chromosome replicates and it attaches right in here, and then this splitting is taking place here through binary fission. And split. It doesn't do prophase, metaphase, anaphase, telophase. It doesn't do any of those steps. Okay? Now, in your mind, think about what is it that you need to focus on most on this. What do you feel the strongest on? What do you feel the weakest on? And make sure you focus on those areas. Don't say everything, because you have some strong things. Tell your bio buddy right now. Go ahead. Okay, now, I hope you had enough time to share. So the first thing we're going to address is just the idea of the cell cycle. This would be a great essay question as well, right? Diagram the cell cycle, bless you. Okay, diagram the cell cycle. Name the different stages. That's not really application level. That's really like a know it, don't know it. That would be the simplest of simple. Some of you, I've talked to you about this when you do your essays. If you can't just diagram the cell cycle and get it right, then you probably cannot do anything where I ask you to apply it. Like, what would happen if you skipped this stage of the cell cycle? How would it be impacted? You see what I'm saying? That would be an application level. For instance, G1. What happens in G1? Growth, and you make more organelles. So if you moved through too quickly through G1, maybe you would know then application level, you would probably be short some what? Organelles. What else happens during G1 that's super critical? The checkpoint. Okay, the G1 checkpoint. What are we looking for in the G1 checkpoint? Did we do, nutrients. Did we do mitosis correctly? Do we want to move forward? Do we have enough nutrients to move forward, just like you did in your POGL? So if we don't do G1, we also miss the opportunity to have a checkpoint. You agree with that? Now, not only do you have a checkpoint where you have to clear security, right? We also learned about this. You not only have to clear security when you go to get on an airplane, you also have to what? Have your ticket. Who's the ticket? Cyclins are the ticket, right? So cyclins build up. It's a checkpoint. You're making more organelles. You're also making proteins, but we talk about a bunch of proteins getting made in the G2, okay, as well. That's the application level of the cell cycle. And we could do the same thing. What happens during the S stage of interphase? DNA replication. What would happen if we skipped the S stage? It's not that you wouldn't have any DNA. I understand what you're saying. You would not have what? Enough DNA, right? You would make cells that were no longer diploid, but in this case they would be what? Haploid. And that's not what you're trying to do in mitosis, because you're trying to make a clone of that cell. Is there a checkpoint during the S stage? Yes. Yeah. You do have a checkpoint in the middle of it to make sure you're going and you're moving forward all right through it. Are you doing it correctly? Okay? The G2 stage. Okay, the G2 stage. What would happen if we didn't do the G2 stage? Not enough proteins for what we needed for the next stage. Okay, so that's what I'm telling you. Just knowing the cell cycle, okay, that's the basics. I expect you to understand that framework, so if I ask you to apply something, you understand it well enough that you can tell me what happens if I have this is elongated, or this is shortened, or this is skipped. Okay? So you told me 2 things on regulating the cell cycle. You have to have cyclins, right? You have to have cyclins, and you have to clear checkpoints. Sorry, just skipped over it. Come back. Here we go. Okay? So we have these checkpoints. Those are the stop signs. Stop. Make sure we're doing that stage okay. Is everything done so far? Or do we need to fix it? And if we can't fix it, what will we do? Kill it. And what is that called? Apoptosis. We also want to verify that we're ready to go on to that stage. That verification, that cell signaling, is cyclins. And remember, we talked about various cyclins at various stages. You see, I know I have it here. There it is. So you can see the different cyclins that have to build up before you go on to the next step. If the cyclins don't build up, you're not going to move on to the next step. What happens if the cyclins stay up and you don't break them down? Is that a problem as well? Yes. Question?
What's the S checkpoint?
Oh, I said S and I meant to say M. Thank you for calling me out on that. I appreciate that. Don't worry about that. I'm sorry. And all of you were like, I couldn't even think what you're saying because I was still thinking about the S checkpoint. I was thinking M's checkpoint. I'm so sorry. Thank you. Okay. What else on this one? I think that's it. Cyclins, cyclins. Okay. Where does the cell spend the bulk of its time? Interphase. In interphase. It spends the bulk of its time in interphase. Okay. When does cytokinesis occur? The very end, but it's also occurring during what? Telophase. I only am bringing that out because it looks like it's 2 separate events, but they really are overlapping one on top of each other. Okay, so if you diagram the cell cycle for me, that's fine. Put it right here, but make a note of it that cytokinesis happens during telophase. Okay, at that point. Okay, good. I think we're okay with that. We talked about this. Yes?
Oh, so if there's the growth in G2, is it proteins growing too, or just more organelles?
Not proteins growing, but in G2 you're making proteins,
yes.
Synthesizing proteins. Yes.
And more organelles too?
Well, the bulk of the organelle formation is going to happen during G1. Yeah. You're going to have overlap on both of those. It's just, the way I think about it is you just underwent mitosis, so you just made 2 cells. And so following that, those 2 cells are going to need to get bigger, and you're going to need to make more organelles to make sure that they can function properly. Okay. What else? Yes?
What stage of interphase are the centrosomes duplicated in?
Well, your centrosomes, now remember what she's saying about centrosomes. Not all cells have centrioles, right? And the centrosome is the region where the centrioles are sitting, or the microtubule organizing center. And you're going to see that early on in prophase. You probably start making the proteins for it in G2, right? But where you're going to see them is when they become visible in prophase. Okay? What else on that? I haven't hit the stages yet. There was one other thing I wanted to talk to you about. Oh, G0. Okay? Why would I enter into G0? What? Okay, yeah, I guess I could be dying, or I don't have enough nutrients. I've done enough replications, right? And so it's basically like an off-ramp, and you're saying, I'm not going to go divide any longer. So you might get big enough just to do your job, and then that's it. You're not going to move forward into the S stage and replicate. Now, I will tell you, there are some exceptions to that rule. There are some cells that kind of enter the G0 stage over here at G2. Okay, but that's an unusual thing where it happens after they've replicated the DNA. But I'm just letting you know that, okay? But that's an exception to the rule. All right, questions on the cell cycle. Okay, let me ask you this. Which one of the checkpoints seems to be really significant of all the checkpoints? G1. G1. Do you remember what gene plays a crucial role right there? P53. Do you remember when we talked about proto-oncogenes and tumor suppressor? Okay, so proto-oncogenes, it's not like they're bad, but remember proto-oncogenes are in a position of what? Power and control. Okay, so maybe they are coding for proteins like the synthesis of cyclins, right? Because if a cyclin increases when it shouldn't, if it was a broken proto-oncogene, what do we call broken proto-oncogenes? Just oncogenes. They are cancer causing. It could be coding for some cell signal that says for the cell to go forward in the cell cycle when it shouldn't. Who normally takes care of those, that missed signal, that wrong signal? Tumor suppressor genes. One of those is the P53, right? And P53 codes for what? Something the friendly ghost? Caspases. And it's the caspases and it ultimately cause what? Apoptosis. So if we're going to bother to know a tumor suppressor gene, let's bother to know a proto-oncogene. Do you remember one of those? P53 is a tumor suppressor. Proto-oncogene is what we want to know. RB, remember? Retinoblastoma. Remember that one? When we talked about it, RAS is like stepping on the gas. Yeah, RAS is like stepping on the gas. And it's telling you to go forward in the cell cycle even when you shouldn't. We will review that, okay? It's just pertinent right here when we're talking about cell cycle regulation. When it can go wrong, we're usually referencing one of those two genes. One is a cell cycle initiator. One is a cell cycle what? Inhibitor. Yeah, it's trying to get a non-initiator. Okay, a stopper. Okay, a stopper if it's out of control. All right. So the signals that we go through, it could be an external signal like a growth signal of some sort. A gland could be secreting it. It could be some sort of hormone that's triggering that cell cycle forward. Or it could be an internal signal that is generated in order to move through. We already talked about checkpoints. Let's talk about these cyclins again. Let's review this. But how about instead of me reviewing it, the person next to you, oldest bio buddy, or no, you can decide however you want. I'll let you pick. One's going to take words, one's going to take the diagram. Okay, remember the diagram? The diagram is here. So decide, you're sitting next to somebody, what are you going to take, the words or the diagram? And then start on the words part. Right, so again, here's another potential essay question. What are the potential essay questions we've said so far? You could just diagram the cell cycle. I could ask you what kind of things promote the cell cycle, what kind of things, what would be the counter to that? Regulate or inhibit. Both promoting and inhibiting are part of regulating, right? I could talk to you about what would happen if you skipped one of those stages, right? Okay, there's that part. Then we could talk about what an example of cell regulation. So we have cyclins and CDKs. You could talk to me about the role of each of these. Which one is an enzyme? CDK, and that stands for cyclin-dependent kinase. Kinase, remember, enzyme. So whoever's turn it is for the picture, you're up. I'll move things. You talk about it. An example of cell signaling is controlling the cell cycle. Good. Needs to build up. Cyclins will build up. Cyclins will bind to CDK. And then it's called a CDK cyclin, which is similar to what we already learned. What did we call that before? Enzyme substrate complex. When you do that, this activates the CDK, so it will transfer A to a protein that was off, turning it on. Or it could take a protein that was on and turn it off. Okay? That is how we can regulate that cell cycle. Okay? That's when we say that cyclins need to build up, we know there's more to the story. When the cyclin builds up, it's activating what? CDKs. Those CDKs are phosphorylating proteins that are turning something on or turning something off. It's the switches. It's the clap on, clap off. Okay? There's a way to control that through cell signaling. All right? And we also learned, we already talked about apoptosis, so I'm going to leave that alone. Next, does DNA always appear as a chromosome, rod-shaped? No. When does it go into the winding stage in order to form a chromosome? Prophase. Remember prophase? The chromosomes become visible. Visible. The chromosomes were there all along, but it looked like something out of your hairbrush, just scattered, chromatin. But it's winding up on those spools. What are the names of the spools that it's winding up on? Histone proteins. And those histones will get together to form what? When you have 8 of them. Starts with an N. Nucleosomes. Speaking of that, have you done your highly suggested reading and thinking yet? I highly suggest it. Okay? Okay? Now, this happens, okay, at what stage again? Prophase. That's when the chromosomes become visible. And when they become visible, the S stage of interphase has already occurred. What happened during the S stage? DNA what? Synthesis or replication. So, it's like it was a mess in there. Now there's 2 messes. And in G2 we still have 2 messes. And then we move into prophase. And they become sister what? Chromatids. Held together by the centromere. It's a temporary holding. When are they going to break apart? What stage? Anaphase. They go away from each other in anaphase, and then it's going to let go. What's organizing this whole dance that we go through? The spindle. Yeah, the spindle. Those microtubules that come out across from the centrosome. Okay? In animal cells, what's sitting in the center of the centrosome? Centrioles. Remember, they look like churros? Remember? But plants don't have centrioles, but they still have a centrosome. They still have microtubules coming out. They still form the spindle. They just don't have the centrioles sitting in the centrosome or microtubule organizing center. Okay? So the whole point of mitosis is to make sure you get a sister in each daughter cell. Okay? So the DNA winds itself and organizes itself in prophase. They line up in the middle, the metaphase plate. During what stage? Metaphase. The sisters go away from each other in anaphase. You reform your nuclear envelope that broke down during prophase. You reform it during telophase. Okay, that's the reforming time. Okay, so I just want us to remember that. Okay, not it. Go ahead, explain it now. All right. Got that part? Okay. So, we talked about diploid and haploid. Okay. Cells that are diploid have 2 copies of every single chromosome. Your body cells, what's another name for your body cells? Somatic cells. Okay? Your somatic cells have 2 copies, a copy from mom and a copy from dad. Okay? Those are called homologous pairs of chromosomes. They are the same size, the same shape, and they code for the same characteristics. This one might say brown hair, this one might say blonde hair, but they're both coding for hair. And there's dominant recessive traits and all of that. But we're going to learn about that in our next unit, and our next unit's on genetics. We'll learn about mitosis, getting ready for sex, and then we'll start talking about the genetics of how that occurs. Okay? And then it'll be the end of the semester, and you'll take a final. Yay! Okay. Now. No. Okay, come back to me. Trying to paint big picture here. In mitosis, my arms, you copy all the chromosomes, you line them all up single file, and you separate the sisters from each other. So every daughter cell has the same chromosomal content as the parent cell. Do you see that? That would be a good essay question. How can you ensure that every daughter cell has the same chromosomal content as a parent cell in mitosis? I gotta remember that. How do you ensure that every daughter cell has the same DNA content as the parent cell when doing mitosis? How do you ensure that? Yes?
For ensuring it, is that the checkpoint where it makes sure
Well, the checkpoint.
the DNA is the same, and then it keeps going and it has the same DNA.
Okay, so you could talk about this question for many aspects. You could just make sure that the life cycle of the cell is going forward and you're not promoting any mistakes by having checkpoints. Okay? But having checkpoints does not eliminate the other ideas, ensuring that I get a sister in each cell. How do I ensure that I get a sister chromatin in each cell? Well, by having them pair up, line up, and separate from each other, right? Through the process of mitosis. Right? Making sure you have the right one without mistakes. Yes, checkpoints. Making sure you do it at the right time, right? You don't want to pull out a cake too early. It's too doughy in the middle, right? So we need what tells us that it's not too early? We have to have a right level of what? Cyclins to tell us ding, right? But we have to have the right ingredients too. We need to put sugar in and not salt, otherwise our cake tastes bad. Who checks? Who's the taste tester for our cake? Checkpoints, right? Checkpoints. How do I make sure that everybody gets an equal slice of my cake? That's the dance of mitosis, to make sure I distribute it accurately. Does that make sense? Of course I have to make it about cake. Okay, now, moving on. We then talked about basically the stages of mitosis. And we talked about how in prophase the chromosomes become visible, this nuclear envelope and the nucleolus inside, that's going to disappear. Okay? We have radiating microtubules. We call that an aster. Okay? These are moving to opposite poles. Okay? And now at this point, everything, the spindle is set up at the end of prophase. My chromosomes are visible. Okay? Then what stage follows prophase? Metaphase. So in metaphase, all the chromosomes are lined up single file. In anaphase, we pull the sister chromatids apart. In telophase, we are reforming the nuclear envelope. That's all nucleus stuff. Prophase, metaphase, anaphase, telophase is all referring to what's going on inside the nucleus. Cytokinesis is division of the cytoplasm. Now, will cytokinesis vary depending on the organism? Yeah. If you're an animal cell, you're going to make a, what's it called? Cleavage furrow. If you're a plant cell, you've got to build a cell wall. Okay, it starts as a plate. The Golgi apparatus is bringing you what you need. What do we have in cell walls? Cellulose. You got to have cellulose delivered, right? So you can build your cell wall. So we can see animal style cleavage furrow, plant cell, cell plate, and then cell wall. All right. We said that mitosis is for growth and repair. We talked about how plant cells will always have meristematic tissue at the ends. They're apical meristems at their shoots and roots at the top to grow out. Some plants can grow in girth. We talked about your highly suggested reading and thinking talks about reproductive cloning versus therapeutic cloning. That's in your reading. Okay? All right. Questions on that? Ask your bio buddy. Got any questions on that? Okay? Come back to me. So then we talked about, and we've already alluded to this partially already, when good cells go bad. Okay? So we talked about that normally cells demonstrate several things. Normal cells have anchorage dependence. They have to be attached to some substratum before they start undergoing mitosis. We talked about normal cells have contact what? Inhibition. But cancer cells don't have either one of those. And we talked about the origins of cancer. We said it could be a problem with one of two genes, right? Proto-oncogenes or tumor suppressor genes, failure to correct DNA mistakes at one of those checkpoints. We talked about telomeres. Do you remember that? How telomerase can make them stay long in the chromosomes. If they're always long, then they will continue to divide. They're like an immortal cell. So we said there's lots of reasons why things can go bad. What does it look like when they go bad? Well, you start to have a neoplasm, you start to have a growth. Now, does that mean you have full-on cancer yet? Only if it becomes what? Malignant. If it gains the ability to spread, okay, in some way. If it goes from being benign to malignant. And we said one of our big concerns is that it gets in either our what or our what? Lymph nodes or our blood vessels, right? And so if it catches and gets into our lymph nodes, that's a problem. Well, what allowed it to stay alive to even gain the mutation to get into one of those vessels? Because normally you'd have all those cells, they'd go hungry. They wouldn't have enough oxygen. What happened?
There was a chemical that made it diverse.
A chemical, do you remember what that was? Angiogenesis. Yeah. Angiogenesis is when the tumor starts to say, come here, little blood vessels, dock into me. Now they have enough nutrients and access to oxygen. They can get rid of their CO2 and waste, and so they can continue to survive.
So is the process called angiogenesis?
Yes, the growing of the blood vessels is angiogenesis, the beginning of blood vessels.
So then what's the chemical?
I didn't say.
Oh.
Yeah. Okay. And then also we started talking about, and this would be a really good essay question, is comparing and contrasting a normal cell with a cancer cell. What are the characteristics of a normal cell versus what you would find in a cancer cell? So we had this slide. We looked at this, where we looked at the structure of the nucleus. Okay? And then we said, and put our focus on two genes. We talked about proto-oncogenes and tumor suppressor genes. So proto-oncogenes, are they bad genes? No, they're just in a position of power, controlling. And these are about promoting the cell cycle and inhibiting apoptosis. They're all about the go, all about the yes, and tumor suppressor genes are about the no. Okay? And so proto-oncogenes, if they mutate into oncogenes, then they're on when they shouldn't be. Okay? They're continuing to produce proteins or cyclins that would promote the cell cycle. And if that happens, hopefully that's not the end of the world, because what would we have to fix that? Tumor suppressor genes who say stop to that. Okay? And then hopefully then nothing happens as a result of that. And why could that happen? Some sort of mutation. Remember, some sort of carcinogen or sunlight that causes that gene to mutate, or you inherited a bad gene from a family member. Okay? And we said that there are over 100 of these. 100 of these genes that are in a position of control. One of them, we said RAS is like stepping on the gas. When RAS goes bad, then it promotes the cell cycle, that protein, because normally RAS would need to be triggered by a receptor who was bound to a growth factor. But in this case, RAS is not. This is poor cell signaling right here. He did not get given the green light, but he's acting like he did, and he's promoting the cell cycle. Okay. And we talked about then tumor suppressor genes, and we talked about P53, remember him? And P53, we talked about that if the P53 is active, okay, then you will go forward with making a protein that will what the cell cycle? Stop. Okay, so P53 builds your brakes. If you don't have brakes in your car, you're going to get in an accident. Even if somebody's stepping on your gas pedal, right? If you lose your brakes, that's when you would end up getting that cancer. And we looked at how P53 operates by preventing you moving forward in the cell cycle and also causing apoptosis. Okay, and I already talked about this, but failure of DNA to repair itself, or telomerase, where your chromosomes keep getting extended, because then that's like an immortal cell. Okay, so that would be another essay question, would be things like, what are the two genes primarily responsible or associated with cancer? And you would name one, name the two categories, give me an example of each, and how they work. Okay, that would be a really good essay question along that line. Okay, and then what do we have last? Oh, that was just a regulation, and we talked about binary fission. Binary fission. This whole unit is about what? What's the topic? Cell communication, right? And cell signaling. So what was the first thing we talked about? What was the first thing you looked at? Homeostasis. You had that chapter 31 where you talked about homeostasis. We talked about positive feedback and negative feedback. What was the next thing we talked about? Immune system, right? Those were examples of cell signaling, right? Are you a part of this body, or are you a foreigner? Do I need to attack and destroy you, or do I support you? You could have two different cells showing the same antigen, but one you help and one you don't help. One you, hey stranger, hi, how are you? That's okay, let me give you a quick hug. We used to work out at the same gym together. Fine. Okay. All right, so one could be promoting an antigen. What's something that could show an antigen and you would help it? MHC2, I will. But name the cell. A presentation cell. Antigen presentation cell. You could have a B could do it, a macrophage could do it, a dendritic cell. Remember these guys? Macrophages. If they present on an MHC2, you will help them. But any other body cell, if you present that same antigen, you're presenting it on an MHC1, and that same signal, the exact same signal, then that causes death, right? Something's going to attack you, and we talked about different ways to do that. We talked about the complexity of the response, whether it's your innate immune response, which we said was like the police force, or special forces. We talked about B cells and T cells, right? All working to wipe it out, either the free floaters or the ones that invaded. What was the next topic you talked about? Flowering plants. Why did we talk about flowering plants? What kind of signals do they receive? The amount of light they have, right? Phototropism. Plants grow towards the light. So what is the signal? The light, right? What is the response? Grow towards the light, right? You talked about photoperiodism, knowing when to flower. We talked about chemical signals that plants could release if they were under duress, right? If you watch that one video, remember cut grass, the smell of cut grass? Remember that? That's not like, yay, summer, cut grass. That's like saying, they're killing me, they're killing me. Right? Did you watch one where it would bring a predatory bug? Right? Those are signaling. You are now talking to a different species with your cell signaling. Do you agree with that? So that's why we went there. What was the next chapter we talked about? The cell cycle. And we looked at signaling within the cell, outside the cell, inside the cell, in order to reproduce. And we talked about how it could go bad, right? Bad signals from bad genes coding for bad proteins, telling us to move forward or stop when we shouldn't. Okay? So that's how it ties it all together, is cell signaling. Now, are we signaling with each other? Am I talking to you?
Yeah.
And you're talking to me?
Yes.
So wouldn't it be a great idea to take a test with a friend, because you could signal back and forth while you were talking and practice cell signaling? Wouldn't that be a great idea? To take a test with a friend?
Yes.
Okay, let's do that. Not the quiz, the test. All right. Look, some of you are like texting. You are my friend. I need to put a caveat, okay? This is my concern. This is my concern, okay? Oftentimes, she's like, I can't even think whatever she says, okay? Okay, this is my concern. I have known all along, remember when Honors Bio got to take a test? I knew you were going to take this test with a partner, okay? I knew it all along, okay? Because I know. Okay, but I didn't tell you. Why didn't I tell you?
So we prepared.
That is the part of me did not want to tell you until the night before the test. You know why? You take your foot off the gas. Exactly. But we have some people. Yes, I got it. But we have some people who have to take the test early because they're going to be gone. So you're like, you can thank them. Okay, so I wanted to tell you today, only so you could start talking about who you want to work with. Now, does this have potential to hurt feelings? Yes. Okay? It does have the potential to hurt feelings. Because you have people, you have friends that hang out in groups of 3, and we've got to be a group of 2, and you've got all of those issues. So you're going to need to navigate this with kindness. Do you hear me? With kindness. And what we will do tomorrow in class is we will make the final decision of who you're going to work with. Now listen to me. Do you think I'm going to make one person work all by themselves? No. Okay, so just take that off the table. That is not going to happen. Okay? But we will work it out. I love you. Make a choice. Yeah, in your class. Okay, have good days.